Okay, good morning. So there's only been 2 things that were requested for me to go over this morning, but if there's anything else that you would like me to go over, I am welcome to do that after I do these 2 things. This is a open review, so anything is fair game. You can ask me any questions that you have, and I will, of course, go over those questions. So the first thing that was requested was to talk about protein folding. So proteins, I'm sure you guys realize this, but proteins are made up of amino acids, okay? And so the amino acid is a couple different components to it. It is going to have a central carbon, and then there's going to be a R group. There'll be a hydrogen, and then there's going to be a carboxyl group, and then there's also an amine group, okay? So let me get those labeled for us. So we have our carboxyl, we have our amine, and then we have, of course, a hydrogen, and then we have this variable R group. And so every single one of your amino acids is going to be the exact same, except for this R group. This R group is going to kind of be different with our 20 different amino acids. So when we're talking about the different folding, we're going to talk about the bonds that are occurring with each of these components, okay? So I'm going to kind of put this little diagram to the side, and I'll keep bringing it out each time that we kind of get to a different level of folding, okay? So our first level of folding that we have is going to be your primary structure. So you'll sometimes see that as like one with a little dot next to it. And if you have any questions during this, you're more than welcome to type them and I can see your questions. So the primary structure is just going to be a string of amino acids. Okay, so it's each of the amino acids are attached to each other. And the bond that we're going to have between these is going to be considered a covalent bond, but we have a more specific name for it. We're going to call it a peptide bond. So the primary structure is just this long string of amino acids, and the bond between it is just a covalent bond. And if you remember, covalent bonds are just bonds in which the two are sharing an electron, and so it could be a polar covalent or it could be a non-polar covalent. If it's a polar covalent, it means that it's not equal sharing, and if it's a non-polar covalent, then they're going to have equal sharing. So then we kind of talk about our secondary structure. Our secondary structure is going to be when we start to see the alpha helix and the beta pleated sheets. And so the bond that we're going to have with this is going to be a hydrogen bond. And so it's important that we understand where these bonds are occurring, right? So in the primary structure, that covalent bond was going to be between the amino acids. So if there's another amino acid here, okay, then we would have that amino group right here, and the peptide bond would be between this carboxyl and the new amino that we would put right on the side. In our secondary structure, we're now talking about the hydrogen bonds. Well, real quick, as a reminder, a hydrogen bond is going to be a bond in which you have a hydrogen attached to an electronegative molecule that's attracted to another electronegative molecule, okay? So what we have is this nitrogen is going to be attracted to this hydrogen that is bonded to this carbon, okay? That is a lie, I'm sorry. the oxygen is going to be attracted to this hydrogen that's attracted to this nitrogen. And so it'll be completely upstream from the structure. So it'll be this really long protein, and upstream from it, it'll kind of fold over. And so you're going to see that your hydrogen bonds are occurring within the backbone. So now our tertiary structure, so the third structure that we can have, this is going to be when we get to our final three-dimensional structure. And so the bonds that you can actually have any type of bond in this. I'm gonna write any bond. Because what's happening is that the bond is going to be between the R groups. And so the R group of one amino acid is going to be bonding with the R group of another amino acid. And so you could have covalent bonds, you could have ionic bonds, you could have hydrogen bonds, you could have van der Waals, you could have just like the hydrophobic interaction in which all of the hydrophobic molecules are all going to bind together. So there's all kinds of things you could have with this one. And again, it's just going to be any bond, and it's between the R group. And then the last thing you could have would be our quaternary, which I never can spell it correctly, so good luck to me. So we could have our quaternary structure. And our quaternary structure is going to be when you have multiple polypeptides. So this has to do with that if I have more than one string of amino acids, this is going to be the interactions, and the bonds, again, can be any bond. And it's occurring between the R groups of different polypeptides. So, do we have any questions on protein folding for the one student that is in the room? We're good? Is there anything else you have questions on in terms of protein folding? Okay, so I guess we'll move over into cell energetics. So with cell energetics, this I believe is what they were wanting me to go over, kind of photosynthesis and cellular respiration, which I went over 2 weeks ago, but I'm sure that the person wasn't in the room at the time. So I'm not going to do the full. I'm just going to kind of do a quick little go over of cellular respiration and a quick little go over of photosynthesis, just to kind of make sure that we're understanding the main components. So cellular respiration has 3 main stages. The first part you have is glycolysis. The second thing we have is, of course, our Krebs cycle. And the third one we have is oxidative phosphorylation. So with glycolysis, it's important that we understand where they take place. So the location of glycolysis is going to take place in the cytosol. So that means that all organisms are able to go through glycolysis because this is a very simplistic step that is taking place in the cytosol, and all organisms have a cytosol, whether it's a prokaryote or a eukaryote, they all have cytosols. And so what's happening in this step is that you have the glucose is becoming pyruvate. So it's being broken down. And so when this happens, there's a couple things that come off. One thing that comes off, we're going to end up with 2 NADHs, and we're going to end up with 2 ATPs, and then we'll also have 2 pyruvates. So I always tell my students it's like a 2-2-2. You have 1 glucose and it makes 2 NADHs, 2 pyruvates, and 2 ATPs. And one thing that's kind of important about this ATP is, how is this ATP made? Well, the ATP is made by something called substrate level phosphorylation. And so with substrate level phosphorylation, that has to do with that at the substrate level, at the actual chemical level, we're going to phosphorylate or add a phosphate to that substrate. And then because of that substrate being phosphorylated, we'll be able to take that phosphate off to make an ATP. And the way that you make ATP is you just take ADP and then you add the third phosphate, which makes ATP, okay? And so overall, we kind of need to understand why these things are important. So the pyruvate is important because it's going to go and move on to the next step. The NADH is important because it's going to move to something called the electron transport chain that we're going to look at during oxidative phosphorylation. So if we move over into the Krebs cycle, in the Krebs cycle, what's going to happen is we're going to go in with something called acetyl-CoA. And it's a cycle, so it kind of goes in a circle. And at this point, we're going to completely break down the pyruvate. Okay, so go completely break down. So we're breaking down the glucose / the pyruvate / whatever form it's in at this point. And so you may be thinking to yourself, well, how did I get from pyruvate to acetyl-CoA? Well, there's another step that I didn't draw in here. And this is what we kind of call that intermediate step, okay? So it's kind of step 1A almost. And this is going to be something called pyruvate oxidation. And so what's happening there is that we're going to lose 1 carbon, and then we're also going to have 1 NADH come off because of the fact that we are breaking down the pyruvate into something called acetyl-CoA, which is how we're able to kind of break it. Like, so the pyruvate becomes the acetyl, we add this coenzyme A to it, which is then able to go into the Krebs cycle because it has that high energy. So within the within the Krebs cycle, 1 turn of the cycle is going to allow us to make a couple different things. We'll make 1 ATP, we'll make 3 NADHs, we'll make 1 FADH2, and then we'll release 2 carbon dioxides. So I always tell my students we have a 3-2-1. So 3 NADHs, 2 carbon dioxides, 1 FADH, and then 1 ATP. And that's, of course, with 1 turn of the Krebs cycle. Since I have 2 pyruvates, I have 2 acetyl-CoAs. And so that actually means that per glucose, I'll go through this twice. And as I said before, it's important that we understand where all these things are going. So the NADH is going to end up moving over here to the electron transport chain. The FADH2 will go to the electron transport chain. Carbon dioxide will be released. And then the ATP is also our substrate level phosphorylation. Okay, oh, and I forgot to mention the location. So the location of this is going to be in the mitochondrial matrix. Okay. So moving on to our oxidative phosphorylation. So oxidative phosphorylation has 2 parts to it. The first part is going to be the electron transport chain. And the second part is something called chemiosmosis. And so what we have going on here is that you've got this membrane, okay? And on the membrane, you have a couple of these things called cytochromes. So cytochromes are just going to be iron-containing compounds that are in the membrane. And what's going to happen is that the NADH and the FADH2 are going to release their high energy electrons. So these high energy electrons are going to get dropped off at the electron transport chain. And so as they're passed from one of the cytochromes to the next cytochrome, they're going to release energy. And so we want to be able to store that energy in some way. And so what ends up happening is that we're going to have the ions are going to move against their concentration gradient. And so we're going to see that at each one of these, we're going to move a hydrogen ion through the membrane. And so there's going to be this high amount of protons on the inside of the intermembrane space. So if this was the IM space right here, and then this is our matrix. So it's going to pump these protons out into that IM space. And so we'll have this high concentration of them, and that leads us to a potential energy. And that potential energy can then be used to fuel the next part of the reaction. So at the end of our electron transport chain, when that electron makes its way down, now it's going to interact with an oxygen, which I don't think you can see yellow. Let me switch to a different color. And I'm running out of space, so how about I just use a pen? So it's going to kind of bind with oxygen. And when it binds with oxygen, it's going to make water. Okay, so we're going to reduce the oxygen. And by reducing the oxygen, plus those electrons, because the electrons is how we're reducing it, plus the protons, gives us that water molecule. Okay. And so at this point, we've made no ATP. So now if we go over into the chemiosmosis step, this is where we're going to use that the high concentration of protons to lead the reaction. And so I have this enzyme right here. And this enzyme is called ATP synthase. And so ATP synthase is the enzyme that's going to allow us to make, of course, ATP or synthesize ATP. So what happens is that the protons fall down their concentration gradient. Okay, we already said there was this high amount of protons up here, right? There's all these protons, too many protons, too many protons. And so as they fall down the concentration gradient, they're releasing that energy. And that energy is then used to make ATP. And so that is the whole part of the chemiosmosis, is that the protons are moving from the IM space back into the matrix, and they're releasing that energy from storage. So do you have any questions at this point to the one student who's sitting in the room? Oh, I didn't mention location. The location is going to be your mitochondrial cristae. So do we have any questions? We're good? Okay, so moving into our next one, which would be photosynthesis. Nope, you're good, Christy. Thanks, Christy, for answering me. So the next thing we have is photosynthesis. So photosynthesis has two steps also. You have the light reactions and then you have the Calvin cycle. So the light reactions, remember we need no location. So the location of the light reactions is in the thylakoid space. I'm sorry, thylakoid membrane. And then the location for the Calvin cycle is going to be in the stroma. So the light reactions take place in, of course, this membrane. So if we have our thylakoid membrane here, there are two of these things called photosystems that sit in the membrane. And so we have photosystem 2 and photosystem 1. Yes, they're named out of order, but that's okay. And in the middle of these photosystems, you've got this reaction center complex that's made up of something called chlorophyll. So that's our reaction center complex. Okay, and it's made up of something called chlorophyll, which is just going to be a pigment, and it's actually chlorophyll A that we have there. So what happens is that the bright, beautiful sun is going to shine down. And when the sun shines down, there are these pigments that are going to be in the photosystem. And what happens is that the light energy is going to activate the electrons of that pigment, and it causes electrons to go to a higher energy state. And so when these electrons jump out to a higher energy state, they've got to pass that energy some way. So if they release the energy back out, it just can make light or heat. But they can also transfer the energy to another pigment. And so what we see here is that the energy is going to get passed from one pigment to the next pigment. And so they're just kind of funneling this energy until they finally get the energy to the reaction center complex. And so when the electrons jump up to the higher energy state from the reaction center complex, then we're going to see that there's going to be this thing at the top called the primary electron acceptor. And what happens is that it actually steals the electron. So my electron is now stolen by my primary electron acceptor. And so there's an electron transport chain in between these two steps. Okay, so at this electron transport chain, of course, the same thing that happened in cellular respiration, we're going to see the electron is going to fall down in energy. And as it falls down in energy, it's going to pump protons out. And those protons will get pumped out into, will get pumped into the thylakoid membrane or thylakoid space. And then we're going to end up making ATP in this process. So I'm just going to write ATP here just so that we can remember that ATP is made in this part. And now you're thinking to yourself, well, this one lost the electrons. Well, how does it get those electrons back? Well, just like we saw in cellular respiration, we made water by reducing the oxygen. Well, now I can oxidize the water, which will release those electrons. And so what we'll see, which I don't have any more colors, we're going to see that right at this point, we're going to have water break down into oxygen, which will release the electrons back to the reaction center complex. And then the electrons that come down this electron transport chain, of course, will go back here. Okay. So the same thing happens with the sun, where the energy passes from one pigment to the next pigment to the next pigment until it finally causes the reaction center complex to lose its electron to the primary electron acceptor. And then it goes down an electron transport chain. And at the end of that, we make something called NADPH. So that is different than the NADH that we saw previously. So NADPH is actually going to be what we use in photosynthesis. And so that's going to be our electron carrier for this process. So there's two different kind of ways that you can move through the light reactions. You can have linear electron flow. And with linear electron flow, it's going to go through both photosystem 1 and 2. And then it's going to make ATP and NADPH. Versus cyclic electron flow is going to deal with only photosystem 1, and it makes only ATP. So what ends up happening with linear is we go completely straight through this process. We have the electron comes up to this primary electron acceptor. It goes down this electron transport chain making ATP. Then it comes back up and it goes down this electron transport chain making NADPH. So we're able to use both photosystem 1 and 2, and we're able to make ATP and NADPH. Versus in the cyclic electron flow, what's happening is that this electron right here just cycles back. So it goes from photosystem 1, goes up to this primary electron acceptor, and then comes down the electron transport chain right here. And so what that does is that allows the energy to just immediately be made only into ATP. And we never really lose our electron because the electron just keeps cycling back through. And so we're going to see when we get into the Calvin cycle that this is important because the Calvin cycle needs more ATP than it does in NADPH. So if we move over into our Calvin cycle, in the Calvin cycle, we're going to use something called carbon dioxide. We're going to release out 1 G3P, and then everything else just gets cycled around. The compound that is going to bind with is something called RUBP. And the enzyme that works here is ribisco. So the carbon dioxide comes in, it binds with RUBP due to ribisco. And then what's going to happen is it's going to go through these couple steps. The first step we have is going to be the fixation. So this is our carbon fixation in which we're going to fixate the carbon dioxide to the RUBP. The second step we'll go through is the reduction. So it's going to gain electrons. So the way that it gains electrons is that we're going to have NADPH is going to drop off electrons. It's actually going to have 6 electrons get dropped off. And then we're also going to have 6 ATPs dropping off energy in order to have this reduction take place, which allows us to make 1 G3P. And then the third step in this is just going to be the rearrangement step. And in rearrangement, we're just going to rearrange the G3Ps, the 5 G3Ps that are still in this process. We're going to rearrange them to make a new RUBP. Or actually, rearrange it to make 3 RUBPs. So there's actually 5 G3Ps still in here. We have 3 RUBPs, and there's 3 carbon dioxides that went in. And in order to do this rearrangement, we're going to need 3 more ATPs. And so that gives you a total of 9 ATPs, but there's only 6 NADPHs, which is why we need to have that cyclic electron flow to make more of your ATP so that you don't run out of NADPH. Okay, so I've gone through the cell energetics and I've gone through the protein folding. What other questions do we have that we need me to go over today? Are there any questions that we need me to go over? We still have 30 minutes that if you want me to go over anything, I'll go over anything. But if no one has any questions for me, then I'll end the live feed. Okay, well, if you have no questions, I hope you have a wonderful day, and let me know if you have questions in the future.